right, pre-AP biology. Today, I want to spend a little bit more time just kind of reviewing some of the concepts we talked about the other day. We were talking about cell transport, and then we're going to continue with that a little bit more as far as how materials get in and out of the cell, which is a really important concept. So, just as a quick review, we talked the other day about the cell, and on both sides of the cell membrane, the inside of the cell, the outside of the cell, there's water. There's solutions of water. It's usually not pure water, but remember, solutions are made up of solvents and solutes. Make sure we know the difference between those terms. We did a simple calculation the other day with concentration, so make sure we can handle that. I talked about the word diffusion, which is the movement of particles from high to low concentration. Again, this is kind of a generic term. You can refer to anything. I think I used the example of air freshener the other day. We talked about iodine in the lab, which moved into the bag from high concentration to low concentration. Again, there's some particles that can easily move across the membrane, others can't. That's because the membrane is semi-permeable. And then finally, I talked about the term osmosis, which is a specific type of diffusion, again, which allows water, specifically water, to move back and forth across the membrane from high concentration to low concentration. So, for example, this animation that I showed you the other day, we can see here's our membrane, and we can see that there's a lot more water on the left than there is on the right, right? So that means that the concentration of water is higher on the left compared to the right. Well, if this membrane is permeable to water, but not permeable to these larger solute molecules, then we would expect that these water molecules would flow in this direction to the right because that's where they're less concentrated at. And that's exactly what happens when I showed you the animation. We see the same thing with this picture. It's basically the same thing. We've got a much higher concentration of water on the left than we do on the right. So that means the water would flow to the area of lower concentration. So make sure we can understand that concept. All right, that leads us to today's notes, which is part 2 of cellular transport. So let's talk a little bit more about cells. Okay, so here's a plant cell. We've got that nice rigid cell wall, around the plant. So you don't see a lot of the organelles in here, like the nucleus and mitochondria, but you do see the little chloroplasts in here, which are kind of large organelles. But anyway, it's a plant cell. You got your chloroplast, you got your cell wall. Now, there's also a cell membrane in there. It's kind of, you don't really see it, but the cell wall and the cell membrane are very close to each other. But just like all cells, things can come back and forth, move across that membrane, because again, the membrane is semi-permeable. It does allow some things to pass through and other things not. So let's talk a little bit about the environment of the cell. What is around the cell? I talked the other day that basically around cells is a solution of water. And what's in that water, like what's suspended in it, solutes, whatever, some of those things can easily transport back and forth, some things can't. But water itself can easily flow back and forth across the membrane. This cell membrane that's around the cell is permeable to water. Okay, so when it comes to the environment, there's basically 3 words that we use to describe the cell's environment. So these are kind of adjective words. All right, the terms are hypotonic, hypertonic, and isotonic. These 3 words describe different environments that the cell can exist in. Okay, so the first term is called hypotonic. It says there's a higher concentration of water outside the cell. So think about a cell. in which the outside, what's on the outside of it, has a higher concentration of water than what's on the inside. So think about which direction water would move. Okay, I'll show you an animation in a few minutes that will help kind of prove to you what happens. Hypertonic is just the opposite. Here we have an environment where there's a lower concentration of water on the outside of the cell. So again, think about where water would move. Would it move in the cell or would it move out of the cell? And then finally, we have something called a isotonic environment, where the concentrations of water are exactly the same on either side of the membrane, both inside and outside. So again, these terms are adjectives. Okay, I want you to remember that. They describe the environment that the cell is in. So here's an example here of a hypertonic environment. In a hypertonic environment, the concentration of water outside the cell is less than what's on the inside. So here we have just a little picture here, and here we have the cell. So let's say on the inside of that cell is 97% water. The other 3% are, solutes or things that aren't permeable. They won't go back. They won't move back and forth. They're just kind of stuck in there unless they move through channels, right? But the inside is 97% water. The outside of the cell, the environment, is 94%, and there's 6% solutes. Okay, but remember, this membrane is permeable to water. It's not permeable to those other things. So we know that water, because of osmosis, water moves from areas of high concentration to low concentration. So just look at the two numbers. Whichever number is higher, okay, 97% on the inside of the cell, that's where it's more highly concentrated and water will leave the cell because it's going to move out to an area of lower concentration. Now, it will move out until it reaches equilibrium. Okay. Now, think about what's going to happen to the cell. If cells lose water, they're going to shrivel, right? They're going to lose water. That's what happens when you put cells in a hypertonic environment. A really good example of this is like if you get into a swimming pool in the summer, or you take a bath or something, right? And like how your skin cells like get all shrivelly, like your skin gets kind of wrinkly. Well, that's because pool water and bath water is hypertonic to your skin cells. So your skin cells actually lose a little bit of water, when you're in pool water or bath water, because that water is hypertonic to your skin cells. If that makes any sense. Now here's kind of just the opposite. Okay, this is a hypotonic environment. In a hypotonic environment, there's a higher concentration of water on the outside of cells compared to what's on the inside. So here we have the same cell, right? 97% water. But look at the environment now. Now there's 99% water on the outside. So again, water moves from high to low concentration. And when it moves into the cell, because that's what it's going to do, the cell is going to swell up. It's going to get larger, right? Now, some cells, if they get, there's too much water, they could explode, right? They could break. Cell membranes are think about like a water balloon. If you fill it up too much, the water balloon can break. Okay, so that could be an issue with cells. We don't want our body cells in hypotonic environments. In fact, we don't really want our cells in hypertonic environments really either. In fact, this is kind of a homeostatic thing. Our body tries to maintain water levels inside your cells all the time. That's one of the functions of your body is to make sure your cells don't have too much water inside and don't have too little water inside. It's kind of a homeostatic mechanism. All right, finally, we have what we call an isotonic environment. Here you've got a cell that's in an environment that has the same concentration of water on the outside than what's on the inside. So this is kind of a cell that is already in equilibrium with its environment, and that's kind of what cells want. They want to have that equilibrium between their environment. Now, just because it's in an isotonic environment doesn't mean that water's not moving back and forth. It's still moving back and forth. It's just moving back and forth at the same rate. So the water that's coming in and the water that's coming out, it's coming out at the same exact rate. And again, that those two concentrations on the inside and the outside are exactly the same. So that's isotonic, which is basically a cell that has met the equilibrium with its environment. Okay. All right, now I want to show you a little animation that oops, that I think represents all three environments pretty well. So here we have some cells. Let's say they're blood cells or whatever. They could be any type of cell. And inside of those cells is a solution of water. Okay, just like your body cells, there's a solution of water in every one of your cells. And let's just say that the concentration is 97% water and 3% salt or some other solute. Okay. So the membranes that are around these cells, again, they are permeable to water. Water can enter and leave the cell. The salt, however, cannot. It stays inside unless it goes through a channel, which is a much bigger process. And we'll talk about that more tomorrow. But if we put these cells into certain environments, think about what's going to happen to these cells. Okay, first of all, this environment on the left, you'll notice that it's 96% water. Don't really even worry about the salt. Just kind of look at the water percentage. So if this is 96 and the inside of the cell is 97, imagine what would happen if you put that cell into this environment. All right, same thing with this cell here. If you put it into this environment, which is 99% water, what would happen to that cell? And then finally, we have an environment that has 97% water in it. Think about what would happen if we put a cell in that environment. So let me play the animation here for you, and you'll kind of see how this works. Okay, so again, there's our cells. We're going to put the first one in. So this is our hypertonic solution over here on the left. Oops, there we go. So you put it in the hypertonic environment, water's going to leave the cell, it's going to shrivel up. You put it into a hypotonic environment, which has more water, that water is going to come into the cell. Cell's going to break, possibly break, doesn't always have to, but it can. Then, of course, we have an isotonic environment. We put that inside, and really kind of nothing happens because there's that equilibrium. All right, so again, we'll watch this one more time. We have a hypertonic environment, a hypotonic environment, and we have an isotonic environment. And I want you to think about what happens to cells when they're in those environments. And I want you to know why. Like, why does a cell get larger, in a hypotonic environment? Why does a cell shrivel when it's in a hypertonic environment? It all has to do with water flow, right? Where is water more highly concentrated, and where is that water going to be moving? Is it going to move inside the cell or outside the cell? Okay, so last couple things here. There's a term here called osmotic pressure. It says it's the pressure that builds up inside of a cell due to a hypotonic environment. So, I mean, imagine a cell that's in a hypotonic environment. Water is coming in, and that osmotic pressure is starting to build up and build up and build up. So, if the pressure is too high, what could happen? I think we saw that in the animation. Now, plant cells and animal cells are adapted a little bit differently to kind of withstand this pressure, right? In, in an animal cell, right, we just have cell membranes. So if the osmotic pressure gets too great in our cells, they're just going to break. Okay. But in a plant cell, there is a cell wall, which does help to withstand some of that pressure a little bit better. So they're a little bit less likely, I guess, to break if they have that cell wall intact. Here's a couple of terms here. I'll move myself over here. There's a term here called plasmolysis. Let's move me over here. Plasmolysis happens when plant cells lose water. For example, I mean, think about what type of environment plant cells would have to be in if they lose water, right? They would have to be in a hypertonic environment. So cells that are in hypertonic environments, they lose water and they tend to shrivel, right? So think about a plant, that like this one here, that kind of looks like it's all droopy and I mean, if those plant cells are in hypertonic environments, they're going to do that. So the term plasmolysis refers to plant cells that are losing water. Okay, so if we kind of think about a plant or even animal cells, but these are kind of specific to plants. I mean, think about the environments that plant cells would have to be in for them to either swell up with water or shrivel, undergo plasmolysis. So, in a plant cell, when plant cells swell, like when their vacuoles get filled with water, that's kind of what's happening. We call that a turgid or a turgid cell. We call this turgor pressure in a plant. I use this word osmotic pressure, but that's kind of a generic term for both plants and animals. In plant cells, we call it turgor pressure. So when those vacuoles get really full, then we can say that there's a lot of turgor pressure inside of those cells, and they're very turgid cells. Whereas if they're in a hypertonic environment, then the turgor pressure decreases and the vacuoles shrivel, and then we call those flaccid cells. Okay, and then think about what a plant would kind of look like, if it had a very high turgor pressure versus a very low turgor pressure in those cells. Okay, so hopefully that makes some sense. All right, guys, that is it for me for today. That's part 2 of cell transport. We will do part 3 in the next lecture video.